Darwin's book, The Origin of Species, was widely read and enormously influential as soon as it was published in 1859. It served to galvanize acceptance of the idea that species evolve. That is, that the biological world we observe today is the result of organic evolution, not an immutable special creation. But Darwin's theory of natural selection, the mechanism he proposed to account for evolution, had a harder time gaining general acceptance. Now, in fact, by the time the 1900s came around, at a time when the scientific community generally embraced the idea of evolution, and at a time when the new field of genetics was beginning to develop, the theory of natural selection that Darwin had proposed had more or less fallen into disrepute. The reason that natural selection failed to gain easy acceptance was because it required Darwin to postulate hereditary processes that he couldn't explain. Somehow parents had to pass traits on to offspring, but Darwin couldn't explain how they did this. So one might think then that when Mendel's work was rediscovered, the beginning of the 20th century, that this would bring about a more general acceptance of Darwin's theory, because Mendel did propose a mechanism of inheritance. Ironically, just the opposite happened. The rediscovery of Mendel's work actually led to greater skepticism about natural selection as being an important mechanism in evolution. Why is this? This is because Mendel's mechanism for inheritance seemed to contradict Darwin's ideas about natural selection. Darwin's views of evolution emphasized gradual changes in traits that showed continuous variation, changes in traits such as the length of a giraffe's neck or the size of a bird's beak, and so forth. But Mendel and the geneticists who rediscovered his work studied the transmission of discrete traits, such as whether a plant had purple or white flowers or a fruit fly had red or white eyes. The hereditary mechanism that was proposed by Mendel appeared unable to account for the kinds of gradual changes in continuously varying characters that Darwin was interested in. As more and more data began to support Mendel's ideas then, geneticists began to assume that natural selection, as Darwin originally proposed it, just couldn't work. Of course, we now know that both Mendel and Darwin were correct. Eventually, the apparent differences between Darwin's view of natural selection and Mendel's theory of inheritance were resolved by what is now called the modern synthesis of evolutionary biology, essentially an intellectual synthesis of ideas that developed gradually over the first decades of the 1900s and really came to fruition in the 1940s and 1950s. There were two factors that were primarily responsible for achieving this reconciliation of the apparent differences between Mendelian genetics and Darwinian selection. The first was this: biologists began to gain a more complete understanding of the potentially very complex relationships between genes and traits, including the realization that the expression of continuously variable traits, such as those that Darwin worked with, almost invariably depends on the contributions of many different genes working together. Second, there was a field that developed to explicitly try to explain how genetic traits could change in populations, a field we now call population genetics, that looked at the transmission of traits not from parents to their offspring, but instead from one generation of an entire population to the next generation of that population. Remember, natural selection acts on the phenotypes of individuals, but evolution occurs in populations. So population genetics provided a formal framework for connecting the principles of Mendelian inheritance with the Darwinian idea of how natural selection could work in populations. In this lecture, we're going to address both of these points in turn as we take a broader look at the mechanisms responsible for evolutionary changes in populations. First, let's quickly look at some of the complexities that can arise when we look in more detail at the relationship between genes and the expression of traits associated with the genes. The key point here really is a very simple one. The relationship between genotype and phenotype is usually not as straightforward as you might think from the discussion of Mendelian traits we've looked at up to this point. Fortunately for Mendel, the traits he happened to study all had a relatively simple genetic basis. Each character was determined by a single gene having only two alleles, with one allele clearly dominant over the other. But in many, if not most cases, we find much more complex relationships between alleles and the expression of trait. Let me outline some of the main reasons that this is the case. The first set of reasons arises from the fact that alleles for a given gene can interact in more complex ways than Mendel had discovered. Consider snapdragon flowers, for example. Flower color in this case is determined by, again, a single gene having two alleles, just as in Mendel's pea plants. Also, like in Mendel's peas, we can find homozygous lines that are true breeding for either red or white flowers. In this case, we'd refer to the red flowers as being homozygous for whatever the red allele was, and white flowers as being homozygous for whatever the white allele is. So far, so good. We're just on the same page as Mendel. However, if we cross these two pure breeding lines of red and white flowered snapdragons, we see something that wouldn't have been predicted by Mendel. And specifically, we find that in the F1 generation, all of the flowers are pink. Now, this seems to contradict Mendel's theory completely. In fact, if anything, it supports the blending hypothesis that Mendel was supposed to have disproved. But if we cross these pink F1 individuals and look at the F2 generation, we get a pattern that begins to become more consistent with Mendel's thinking. Among the F2 offspring, we find a phenotypic ratio of one red individual for every two pink individuals for every one white individual. So a 1 to 2 to 1 ratio of red to pink to white. These phenotypic ratios are the same as the genotypic ratios we would have expected from a typical monohybrid cross. And we can conclude from them that what's happening is that the heterozygous genotype has its own unique phenotype. It's not that the red allele is dominant over the white allele or vice versa. When a red allele and a white allele occur in the same heterozygous individual, the phenotypic result is a blend of the two. We call this circumstance incomplete dominance. Now, the relationship between genotype and phenotype can become even more complicated than this when we look at cases where there are more than two alleles for a particular gene. In fact, cases where there are only two alleles, such as those that Mendel worked with, are relatively rare. Usually genes have several, sometimes dozens, or maybe even hundreds of different alleles in a population. Any individual can have only two alleles total, but they can have many different possible combinations of two alleles. A simple example of a trait that has multiple alleles is the so-called ABO blood group system in humans, which also illustrates an interaction between alleles called codominance. There are three types of alleles in this case. Let's call them the A allele, the B allele, and the O allele. A and B are both dominant over O, but A and B are not dominant over each other. In fact, they're codominant. What that means, if the A allele and the B allele occur in the same individual, they both express themselves. So what you get is, if you're a genotype that has two A alleles, or an A allele and an O allele, you will have the phenotype of blood type A. Or if you've got two B alleles or a B and an O allele, you'll be blood type B. If you have an A and a B allele, then you're blood type AB. You have what are actually cell surface proteins or associated with your blood that are expressed by both of these two genes, both of these two alleles. If you're homozygous for the O allele, then you will be the relatively rare, or the relatively common, I should say, blood type O. Now, interactions among alleles actually can be far more complicated than these relatively simple examples, but they serve to show us that we have to move beyond Mendel when we think of the relationship between genotype and phenotype, even when we're only looking at one particular gene. Now, another factor that complicates the relationship between genes and traits is that different genes often interact with each other. For example, the phenotypic expression of one gene often is affected by another completely different gene in a condition that we call epistasis. Epistasis comes from the Greek meaning causing to stop, because often the interaction involves the expression of one gene simply blocking the expression of another. Consider, for example, fur color in mice. In this case, there's a gene which happens to have two alleles that affects the darkness of the fur's pigment, with the allele for black coat color being dominant over the allele for brown coat color. Mice that are homozygous for the black allele or heterozygous will have potentially black fur coats, and only mice that are homozygous for the recessive brown allele can be brown. This seems simple, but there's more to the story. There's an entirely different gene that affects whether the pigment gets deposited in the hair in the first place. Now, if the mouse has a genotype at this second allele, or at the second locus that prevents pigment deposition, then the mouse will be white, regardless of what its genotype is for fur color. In this way, there's an epistatic effect of pigment deposition on fur color. The phenotypic effects of the fur color gene are not independent of a completely different genetic locus. Now, there's a third and really ubiquitous set of factors that affect the relationship between genotype and phenotype and make it much more complex than the kinds of relationships that Mendel worked with. And this is the effect of the environment on how genes are expressed. In general, there are few, maybe no cases where the phenotypic effects of a gene are not influenced potentially in some way by the physical environment in which an organism finds itself. Even in Mendel's experiments, the full expression of phenotypes such as flower color or seed shape and so forth were affected by environmental factors such as the amount of sunlight the plants received, the amount of water or nutrients and so forth that they had available to them. Mendel understood this, and in fact he took great pains to raise plants under identical physical conditions as much as possible to remove these environmental effects, and that's why he was able to see relatively clear dichotomous traits. In the real world, of course, the environments that individuals with different genotypes find themselves in is not controlled in this way, and therefore environmental factors generally have a very strong effect on gene expression. We're going to look in more detail at how the environment affects gene action later in the course when we talk about development. Now, the final reason I'll cite for why the relationship between genes and traits is more complex than Mendel and his followers originally imagined is the fact that for many heritable traits, the expression of the trait depends on the action of many different genes in a cumulative fashion. Mendel, as you know, worked with discrete traits, but many of the most interesting traits, including those that Darwin emphasized, don't sort out into discrete categories, but instead show continuous variation along some continuum. We call these kinds of continuously varying phenotypic characters quantitative traits. They're called quantitative traits because the phenotype of an individual has to be measured with some sort of ruler or yardstick, or quantified, we'd say, on some continuous scale. Quantitative traits share in common the fact that they're almost invariably the product of the additive effect of alleles across several, if not many, many different genes. And that cumulative effect is what contributes to a single phenotypic character. Let me make this clear with an example. Skin color in human is a quantitative trait with a continuous distribution of skin colors ranging from very light, of course, to very dark. There's evidence that skin pigmentation is affected by at least 3 independently inherited genes. Now, the real story is a little more complicated than this, but I'm simplifying here to make a point. Each of these genes has 2 alleles, one associated with the production of more skin pigment and one associated with the production of less skin pigment. The effect of the alleles for all of these genes is additive. That is, the more of the dark skin alleles you possess for any of these 3 genes, the darker your skin. So a person with 6 dark-skinned alleles across all 3 genes, which is the total possible you could have, would have the darkest skin, whereas an individual with no dark-skinned alleles would have the lightest skin. And of course, individuals that have some number of alleles in between would have intermediate skin colors. So in this simplified example then, individuals would at least range along a set of categories ranging from 0 skin alleles to 6 skin alleles. In other words, at least 7 gradations. Now, in fact, this greatly oversimplifies that particular case because there are many other factors, including genetic factors, that affect skin color. But it makes the general point that as we add more genes affecting a single trait, we get a more and more finely divided continuous gradation of the possible phenotypes that an individual can express. And it's this continuous variation that was what Darwin wanted to explain with his evolutionary theories. It was the realization that quantitative traits could be actually accounted for by the action of multiple genes, each of which on its own conformed to Mendelian principles of inheritance, that was one of the major contributors leading to the synthesis of Mendelian genetics with Darwin's ideas about natural selection. Okay, now let's turn our attention to the other thing that contributed in a major way to reconciling the work of Mendel and Darwin, which was an understanding of how to account for the transmission of traits not just between parents and offspring, but across successive generations of entire populations. Because evolution occurs on the level of populations, we need to be able to describe genetic variation in population terms, not just in terms of the genotypes of individuals, in order to understand how that variation is maintained or changed over time. That is, what we want to do is to understand how alleles for particular genes are transmitted in populations as a whole. To do that, let's first define what we might want to call a population. A population, in genetic terms, is a localized group of individuals of the same species that all are capable of interbreeding. They're in the same place, they're the same species, they can all mate with each other. Because these individuals can interbreed, we can consider their genetic makeup in an aggregate fashion. We call this aggregate genetic makeup the gene pool of a population. The gene pool is literally all of the alleles for all of the gene loci for all of the individuals in a population. When we were working with Mendelian genetics, the possible genetic makeup of an offspring depended solely on the particular alleles present in the genotypes of its parents. But in population genetics, the genetic makeup of a future generation in a population is drawn from the gene pool as an aggregate whole and depends on the relative frequencies of all alleles available in that population, potentially regardless of which individuals have those alleles. In the most fundamental terms, then, population genetics is going to allow us to represent evolution as a change in the gene pool of a population, which ultimately has to be what's happening. The genes, or the alleles for genes, have to be changing for the phenotypic characteristics of a population to evolve. Let's consider a concrete example for how we can describe allele frequencies in a population gene pool. I'm going to pick a relatively simple and fun trait to consider. Ask yourself if you can roll your tongue. That is, see if you can stick out your tongue and roll the sides upwards. Now, it turns out that this trait, almost remarkably, seems to be determined by a single gene that has two alleles. People who can roll their tongues have the dominant phenotype, meaning they have at least one copy of the dominant allele. Let's call it big T. They might be homozygous or they might be heterozygous. And individuals who can't roll their tongues are homozygous for the recessive allele, little t, little t. Now, if we sample 100 individuals in a typical human population and count how many people can and cannot roll their tongues, what we find is that 91 people on average can roll their tongues and 9 on average can't. Now, actually the numbers are a little different from this. But the fact is that the non-tongue rollers are the rare phenotype. And I'm using those numbers because they'll help things work out more evenly later on. The 91 individuals who can roll their tongues, or I should say 91% of the population, and the 91% of the population that can roll their tongues, and the 9% of the population that can't, represent the frequencies of the phenotypes in the population for this trait. But what does the gene pool for this trait look like in a population? Another way to put this question is, what's the relative frequency of the two alleles for this trait in the population as a whole? So if we have this population we've sampled of 100 people, one thing we know is that there are 200 alleles. Every individual has 2 alleles. If we've got 100 people, we've got a gene pool for this one trait. Now, we're just talking about the one trait. That's 200 alleles. The question then is, of those 200 alleles, what percentage are big T alleles and what percentage are little t alleles? Now, it's hard to answer that question directly, because although we can count the phenotypes in a population by just asking people if they can roll their tongue or not, we can't determine directly the genotypes. And that's because although we know the non-rollers are both little t, we don't know what the proportions of rollers are that are homozygous or heterozygous. Now, if we did know that, what we could do is then just count up the number of alleles total. Let me just simplify the math and tell you that because we know the frequency of the recessive phenotype, we can do some back calculations and figure out what the relative frequencies of the homozygous dominant phenotype and the the homozygous dominant genotype and the heterozygous genotype must be. As I said, these can be tongue twisters. And I'm going to spare you the details and just tell you that if we do the math, we would find the following allele frequencies in a population like this. 70% of those total 200 alleles would be big T alleles, that is 140 on average out of 200, and 30% would be little t alleles. Let's just call, for the sake of argument, the frequency of the big T allele P and the frequency of the little t allele Q. Typically, we refer to these frequencies when we're talking about frequencies of alleles in gene pools in terms of decimal fractions. So we would say that P, the frequency of big T, is 0.7, and Q, the frequency of little t, is 0.3. Because there are only 2 alleles in the whole gene pool in this example, the total has to equal 1. In other words, P plus Q has to equal 1. Now, let's say we've described the gene pool of the population in this way. What can we do with this information? The question we want to ask next, and this is really getting to the key question, why we're doing this exercise, is what would happen to these allele frequencies, P and Q, if this population mated and produced another population? In other words, if all the individuals in this population reproduced and we got another generation of this population. This is an important question to begin asking because remember, evolution represents a change in relative allele frequencies in a population's gene pools. So we want to ask, how can we look at how these allele frequencies change? A simple mathematical model for answering this question was introduced actually by 2 different people in the same year, in 1908, by G.H. Hardy, who was an English mathematician, interested in probability theory, and Wilhelm Weinberg, who is a German physician interested in population genetics as an amateur. Now, Hardy and Weinberg independently reasoned that we could model the mating of a population by considering picking alleles from that population's gene pool at random. A way to think about this is to imagine that the gene pool of a population is like a big bucket of alleles. Each time 2 individuals mate, it's as though you reach into that bucket, pick out one allele, which would be a gamete from one individual, then reach in again, pick out another allele, which would be a gamete from another individual, and join them. And that would be a new individual in the next generation, and you throw those 2 alleles in another bucket, which represents the gene pool of the next generation. Now, when you do this, it's relatively easy to figure out the relative frequencies of genotypes of the offspring that would be produced. And that is because as you draw these alleles out at random, you're going to be drawing alleles out in the same proportion that they occur in the bucket. So for example, in our example, for example, because 70% of the alleles in the gene pool are big T, you have a 70% chance of pulling out a big T allele. And of course, you have a 30% chance of pulling out a little t allele. If you just close your eyes and reach in, your probability of pulling out an allele will depend on the relative frequencies of the alleles in that gene pool. Now, here's the simple trick that both Hardy and Weinberg introduced. The likelihood of producing an individual with the homozygous genotype big T, that is the joinder of 2 big T alleles, is the joint probability of drawing first one big T allele and then another big T allele, which is 0.7 times 0.7, or 0.49. Similarly, the likelihood of producing an individual with the homozygous recessive genotype, little t, is 0.3 times 0.3, which equals 0.09. Calculating the total frequency of heterozygous individuals having big T and little t genotypes is slightly different because we can get these individuals in two ways. You could first draw a big T and then a little t, or you could draw a little t and then a big T. So that means that the frequency of the heterozygotes in the next generation will be 2 times the joint probability of drawing a big T and a little t allele, which is 2 times 0.7 times 0.3, which equals 0.42. Now, I don't expect you to follow this math in your head, although you could work this out on a piece of paper if you wish. So let me tell you what we've just done, following the approach taken by Hardy and Weinberg. We used the known allele frequencies in a gene pool to calculate the relative proportions of individuals having the 3 possible genotypes for this trait in the next generation of the population. Using the names that we've given to the frequencies of these allele types, we'd say that there are p squared homozygous big T individuals, q squared homozygous little t individuals, and 2pq heterozygous individuals. Just as for the allele frequencies, these genotype frequencies have to add to 1. So in other words, p squared plus 2pq plus q squared equals 1. Using the allele frequencies we started with, this means then that 49% of all offspring will be homozygous dominant, 42% will be heterozygous, and 9% will be homozygous recessive. Notice that if we look at the phenotypic frequencies associated with these genotypic frequencies, we would find 91% of all individuals to have the tongue rolling phenotype because we would be adding the 0.49 and the 0.42% of the homozygous dominant and the heterozygous, which is where we started at the beginning. More importantly, and this is where we get to the crux of the matter, we can ask what the allele frequencies are in this next generation that we've just produced by sexually reproducing our bucket of alleles this way. I won't take you through the math here, but suffice it to say that the way you would do this is simply to count up all the alleles that must be found in those individuals having the genotype frequencies we just calculated. So for example, each homozygous dominant individual must have 2 big T alleles. So we ask how many proportionately homozygous individuals there are, multiply it by 2, and that's some of the big T alleles, and then we add to that the half the number of heterozygous individuals, because each of those heterozygotes will have 1 big T allele, and so forth. If we do this kind of math, what we find is that after we've gone through a generation of sexual reproduction, we end up with the frequency of the big T allele in the next generation as 0.7, and the frequency of the little t allele in the next generation is 0.3. But these are exactly the same frequencies of alleles we had in the first generation. We started with 0.7 and 0.3. We allowed this population to breed randomly, to sexually reproduce and create a new generation, and we find that we have the same genotype frequencies as we began. Nothing has happened to the allele frequencies in this population from generation to generation. This result we call the Hardy-Weinberg equilibrium. It's an equilibrium because it suggests that allele frequencies and genotype frequencies will remain constant in a population from generation to generation. Now, this is a little surprising because we're interested in how allele frequencies change, how gene pools change. But the Hardy-Weinberg calculations we've just gone through show that if we allow a population to reproduce sexually, we don't expect anything to change at all. This surprising conclusion raises really 2 questions. First, what happened to all that variation that was supposed to be created by sexual reproduction? I've been going on and on about how sex creates genetic variation. We've just had a population go through sexual reproduction and we get the same allele frequencies as we had before. The answer is that genetic recombination has no effect on allele frequencies. All it does is just shuffle the relative combinations of alleles. In other words, meiosis shuffles the genetic deck, but it doesn't change the cards you've got in the gene pool. The second question I want to ask is this: how can we ever get a change in allele frequencies in a population if this Hardy-Weinberg equilibrium tells us they generally don't change? The answer to this second question has to do with the fact that the Hardy-Weinberg equilibrium only holds if certain assumptions inherent in the mathematics that underlie it are met. when those assumptions are violated, allele frequencies will change from one generation to the next. And it's by understanding what those assumptions are and how they're violated that we gain insight into the mechanisms that are responsible broadly for evolutionary change in populations. And that's going to be the subject of the next lecture.